Hi friends, welcome to the Knowledge Club Online. Now I am going to discuss about the gene linkage. On the basis of his studies of the garden peas, means the Mendel's studies of garden peas in the 1850s, George Mendel formulated some of the fundamental principles concerning the mechanisms of heredity. First, he deduced that the units of inheritance are discrete, occur as pairs, and can exist in alternative forms. Later, by about 1905, the units of inheritance were called genes by Johanson, and each variant of a gene was called an allele. Secondly, Mendel found that only one member of each pair of genes is distributed to a sex cell or gamete, that is, the gametes are always pure. Thirdly, he concluded that pairs of genes assort independently of one another, with the result that all possible genetic combinations will occur if enough offspring from a single genetically appropriate mating are sampled. It can be explained by taking following example. If two genes are on different chromosomes, first one, this is the interphase of chromosomes are unpaired. That is the prophase phase and the cell division. The chromosomes and centromeres have replicated, but centromeres have not split. This is the prophase homologous synapse. This is the anaphase centromere attached to spindle and are pulled to pores of cell. The other equally frequent alignment. This is the telophase, two cells form. The second anaphase, new spindles form and centromeres finally divide. And at last, the end of meiosis, four cells produce from each meiosis. The total 1/4, 1/4, 1/4, 1/4. Now, if two genes are on different chromosomes, this is the parental chromosome, the gametes are AB, small ab, the mitotic diploid F1 generation produce this. The half look like they got a set of parents' chromosome after the F1 generation, and half look like they got a mix of both parent chromosome, that is the recombinant style. Now, suppose both genes A and B are next to each other on the same chromosome. This the interphase chromosomes are unpaired, and prophase chromosomes and centromeres have replicated, but centromeres have not split. The prophase analogs, what happens to the ratio in this diagram? The meiosis with no crossover between genes, the all parental chromosomes are produced, but the meiosis with a crossover between the genes, that is the two parental and two recombinant chromosomes are produced. In this case, the two gametes form the F1 generation, the two gametes, and after the F2 generation test cross progeny, the 1/4 condition of a parental 2 and 1/4 combination of recombinant chromosomes are produced. Now, the genetic recombination and linkage. The genetic findings of Mendel and Morgan related to the chromosomal basis of recombination. But genes characterizing two different traits are not always present on two chromosomes. When two or more genes reside on the same chromosome, they are said to be linked, and their transmission pattern is called linkage. They may be linked together on the autosomes or on the sex chromosome. Genes present on different and non-homologous chromosomes are called unlinked genes. Unlinked genes assort independently of one another during the formation of gametes, Mendel's law of independent assortment, Linked genes on the same chromosome, however, tends to stay together during the formation of gametes. Thus, the results of test cross and self cross involving two genes yield differing results depending on whether the genes are linked on same chromosome or unlinked on different chromosome. How linkage affects inheritance? Morgan did other experiments with fruit flies to see how linkage affects inheritance of two characters. Morgan crossed flies with differed in traits of body color and wing size. This is the two fruit flies. The gray body one is a gray body normal wings and another is black body vestigial wings. When F1 generation test cross is produced, after the F1 generation test cross, it reproduced the test cross offspring, the wild type grey normal, black vestigial. Grey vestigial and black normal. The predicted ratio, if genes are located on defined chromosomes, 1:1:1:1:1 is produced. But if genes are located on the same chromosome and parental alleles are always inherited together, that is the 1:1. At the result, the 965 wild type grey normal is produced, then the 944 the black vestigial produced, and 206 number of grey vestigial produced, and 185 the black normal fruit flies are produced. Morgan found that body color and wing size are usually inherited together in specific combination parental phenotypes. He noted that these genes do not assort independently and reasoned that they were on the same chromosome. F1 dihybrid female and homozygous recessive male in test cross, the offspring produce this. However, the non-parental phenotypes were also produced. Understanding this result involving exploring genetic recombination, the production of offspring with combination of traits differing from either parent. This lecture may be helpful to you. Thank you.